Welcome, dear listener. Find yourself a comfortable position, perhaps lying down in your favorite spot where the world feels soft and distant. Let the weight of the day begin to dissolve as you settle into this quiet moment. Take a slow, gentle breath, feeling your chest rise and fall like waves on a calm sea. Tonight, we embark on a journey through time, exploring one of humanity's most visible and fascinating evolutionary stories. Let your mind drift with me as we unravel the science, the history, and the beautiful complexity behind the spectrum of human skin colors that paint our world today. Close your eyes if you wish, and let these words carry you into a deeper understanding of who we are and how we came to be. Human skin color is an evolutionary adaptation to varying levels of solar ultraviolet radiation in different geographic regions. To truly appreciate the story of human skin color, we must first understand what skin actually is and how it interacts with the light that bathes our planet every single day. Imagine, if you will, the largest organ of your body, this remarkable barrier that stands between your internal world and everything external. Your skin is not merely a covering but a sophisticated interface, a dynamic ecosystem that has been shaped by millions of years of evolutionary pressure. The color of human skin arises from a pigment called melanin, a term derived from the ancient Greek word for black or dark. Melanin is not a single substance but rather a family of complex biomolecules, organized as polymers that form within specialized cells called melanocytes. These melanocytes reside in the basal layer of your epidermis, the outermost layer of skin, and they are the artists responsible for painting each human being with their unique shade. There are two primary types of melanin that concern us in this story. The first is eumelanin, which comes in two variants, black eumelanin and brown eumelanin. This pigment provides those rich, dark browns and deep blacks that we see in skin, hair, and eyes. Eumelanin is a photoprotective champion, absorbing and neutralizing harmful ultraviolet radiation with remarkable efficiency. The second type is pheomelanin, which produces the reds and yellows we observe in lighter skin tones, freckles, and red hair. Unlike its darker cousin, pheomelanin offers far less protection from ultraviolet radiation and can even become unstable when exposed to sunlight, potentially contributing to cellular damage. The ratio and total amount of these two melanin types, combined with the number, size, and distribution of melanosomes, the tiny organelles that house melanin, determine the incredible diversity of human skin tones we see across our species. In individuals with deeply pigmented skin, melanosomes are larger, more numerous, and packed densely with eumelanin. The pigment is distributed not just near the surface but throughout the deeper layers of the epidermis, providing comprehensive protection. In contrast, those with lighter skin have smaller, fewer melanosomes containing varying mixtures of eumelanin and pheomelanin. Fascinatingly, research has shown that regardless of skin tone, the ratio of eumelanin to pheomelanin in human epidermis remains relatively constant at approximately 74% eumelanin and 26% pheomelanin. What changes dramatically, however, is the total amount of melanin produced. This discovery reveals that skin color diversity is not about fundamentally different biological systems, but rather about the regulation of melanin production, a dimmer switch rather than an on-off mechanism. Now, let us turn our attention to the force that has sculpted this diversity: ultraviolet radiation. The sun, that brilliant star that makes life on Earth possible, emits a spectrum of electromagnetic radiation. A portion of this spectrum falls in the ultraviolet range, invisible to human eyes but profoundly influential on biological systems. Ultraviolet radiation is divided into three categories based on wavelength. UVC radiation, with the shortest wavelengths from 200 to 289 nanometers, is the most energetic and potentially dangerous. But fortunately, Earth's ozone layer absorbs it entirely before it reaches the surface. UVB radiation, spanning from 290 to 320 nanometers, is less energetic but still powerful enough to cause significant biological effects. It penetrates the upper layers of skin but is largely absorbed before reaching the dermis. This is the radiation that causes sunburns, damages DNA directly, and paradoxically, triggers the synthesis of vitamin D in our skin. Finally, UVA radiation with wavelengths from 320 to 400 nanometers is the least energetic but most abundant, comprising about 95% of UV radiation reaching Earth's surface. UVA penetrates deeply into the dermis and generates reactive oxygen species that can cause indirect DNA damage and premature aging. The interaction between UV radiation and human skin is a complex dance of damage and defense, destruction and synthesis. When UV photons strike skin, several things happen simultaneously. DNA absorbs UV radiation, particularly UVB, which can cause direct damage by creating thymine dimers, abnormal bonds between adjacent DNA bases that distort the double helix and interfere with replication. If these errors are not repaired, they can lead to mutations and potentially cancer. UV radiation also generates reactive oxygen species, unstable molecules that damage cellular components including lipids, proteins, and nucleic acids. These reactive molecules are like tiny molecular hand grenades exploding within cells. However, UV radiation is not purely destructive. UVB radiation in particular performs an essential function. It drives the synthesis of vitamin D. When UVB photons penetrate the skin, they interact with a molecule called 7-dehydrocholesterol present in skin cells, converting it into pre-vitamin D3, which then undergoes further transformations to become active vitamin D. This vitamin is crucial for calcium absorption, bone health, immune function, and numerous other physiological processes. Herein lies one of evolution's great balancing acts. Too much UV radiation damages and destroys, while too little deprives the body of essential vitamin D. Melanin acts as nature's sunscreen, absorbing and scattering UV radiation before it can cause harm. The effectiveness of this protection depends on the type and amount of melanin present. Eumelanin is particularly efficient, absorbing UV photons and dissipating their energy as harmless heat. Studies have shown that melanin can absorb up to 99% of UV radiation. Additionally, melanin functions as an antioxidant, scavenging reactive oxygen species before they cause damage. The melanosomes containing melanin are strategically transferred from melanocytes to surrounding keratinocytes, the predominant cells of the epidermis, where they position themselves above the cell nuclei like tiny protective umbrellas, shielding DNA from radiation damage. The amount of melanin in skin is not static. In response to UV exposure, skin tans, a phenomenon called facultative pigmentation. This involves two mechanisms: immediate pigment darkening, which occurs within minutes of UVA exposure and appears as a grayish or bluish coloration, and delayed tanning, which develops over days following UVB exposure as melanocytes increase eumelanin production. This tanning ability varies greatly among populations and represents an adaptation to environments with seasonal variation in UV intensity. Understanding these fundamentals, the types of melanin, the nature of UV radiation, and their intricate interactions, provides the foundation for appreciating the remarkable evolutionary story that unfolds across hundreds of thousands of years of human history. This is not merely a story of colors and chemicals, but of adaptation, survival, and the endless ingenuity of natural selection responding to environmental pressures. To understand why humans display such extraordinary diversity in skin color, we must travel back in time, deep into our ancestral past, to the sun-drenched landscapes of Africa where our species first emerged. The story of human skin color is inextricably linked to the story of human migration, one of the most dramatic chapters in our evolutionary history. The scientific consensus, supported by extensive genetic, fossil, and archaeological evidence, indicates that anatomically modern humans, Homo sapiens, arose in Africa between 300,000 and 200,000 years ago. The exact location remains a subject of ongoing research, but evidence points to regions in East Africa, possibly near the Great Rift Valley or what is now Ethiopia and Kenya. These early humans lived in environments characterized by intense solar radiation throughout the year, particularly in equatorial and near-equatorial zones where the sun passes nearly directly overhead. But the story of human skin actually begins even earlier with our pre-Homo sapiens ancestors. Between 2 and 3 million years ago, early hominins underwent a dramatic transformation. They lost most of their body hair. The reasons for this hair loss remain debated, but leading theories suggest it was an adaptation for thermoregulation, cooling the body during extended periods of physical activity in hot environments, particularly as early humans adopted more active hunting and scavenging strategies involving long-distance travel and running in open savanna grasslands. This hair loss exposed skin directly to intense tropical sunlight, creating powerful selective pressures. Recent genetic studies have revealed something surprising about our ancestral skin color. Before losing their body hair, our ancestors likely had light skin beneath their fur, similar to modern chimpanzees, our closest living relatives. When you look at a shaved chimpanzee, you see pale skin protected by a dense covering of hair. But once that protective hair covering was lost, everything changed. The development of dark, melanin-rich skin in early humans was likely a rapid evolutionary response to the harmful effects of intense UV radiation on exposed skin. Genetic evidence suggests that deeply pigmented skin evolved in the genus Homo within the past 1 to 2 million years. This timing coincides with the loss of body hair and the emergence of more complex hunting and gathering behaviors that kept our ancestors active in open environments under direct sunlight for extended periods. For hundreds of thousands of years, all humans were darkly pigmented, living exclusively within Africa. Dark skin was not merely advantageous but essential for survival in equatorial environments. The melanin-rich skin of these early Homo sapiens absorbed and scattered the potent mixture of UVA and UVB radiation that bathes tropical regions year-round, protecting against DNA damage, preventing the photodegradation of folate, maintaining skin integrity, and allowing humans to remain active during daylight hours without suffering debilitating sunburns or long-term tissue damage. Then, beginning around 120,000 to 70,000 years ago, something momentous occurred. Some human populations began leaving Africa. This migration out of Africa was not a single, simple exodus, but rather a complex series of movements involving multiple waves of dispersal, some of which ultimately failed while others succeeded in establishing lasting populations across the globe. The first significant migration led to populations moving into the Middle East and the Arabian Peninsula around 70,000 to 60,000 years ago. These populations encountered environments different from the African heartland, regions with more seasonal variation in daylight and UV intensity, though still relatively sunny. As humans spread from these initial beachheads, they followed various routes. Some moved eastward along the southern coast of Asia, eventually reaching Southeast Asia, Australia, and the Pacific Islands. Others moved northward into Europe and Central Asia. Still others traveled across Central Asia into East Asia and eventually across the Bering Land Bridge into the Americas. Each of these migrations exposed human populations to new environments with different UV radiation regimes. The further from the equator populations traveled, the more dramatic the changes in their UV environment became. At higher latitudes, the sun's rays strike Earth at more oblique angles, traveling through more atmosphere and thus delivering less UV radiation to the surface. Additionally, these regions experience dramatic seasonal variations, long, dark winters with minimal UV exposure alternating with brighter summers. Archaeological and genetic evidence reveals that these migrations were not uniform or continuous. Populations moved in fits and starts, sometimes expanding rapidly, sometimes remaining in particular regions for thousands of years, sometimes retreating or dying out entirely. The archaeological record shows that humans reached the Levant by at least 100,000 years ago, Southeast Asia and Australia by 60,000 to 50,000 years ago, Europe by 45,000 to 40,000 years ago, and the Americas by at least 15,000 years ago, though possibly much earlier. As these populations dispersed across the globe, they carried with them the dark skin pigmentation of their African ancestors. But in many regions, this ancestral pigmentation was no longer optimal. In environments with reduced UV radiation, deeply pigmented skin, while still protective against what UV radiation was present, began to pose a different problem. It inhibited the production of sufficient vitamin D. This created new selective pressures, and natural selection began favoring genetic variants that reduced melanin production, leading to the gradual evolution of lighter skin tones. Crucially, this depigmentation did not occur through a single mechanism or in a single population that then spread these genes around the world. Instead, genetic studies have revealed that lighter skin evolved independently multiple times in different regions through different genetic pathways. The genes responsible for light skin in European populations are largely distinct from those producing light skin in East Asian populations, providing powerful evidence of convergent evolution. Different populations arriving at similar solutions to similar environmental challenges through different genetic routes. For instance, mutations in the SLC24A5 gene, which dramatically reduces melanin production, swept through European populations within the past 10,000 to 30,000 years and is found in extremely high frequencies among Europeans today. However, this same variant is found at much lower frequencies or is absent in East Asian populations, who instead evolved light skin through different genetic changes, including mutations in genes like KITLG and MFSD12. The rate of these evolutionary changes varied dramatically depending on the strength of selection pressures. In extreme environments, the far northern reaches of Europe and Asia, where winters are long, dark, and cold, selection for lighter skin was particularly intense, and genetic changes spread rapidly through populations. In intermediate latitudes, where UV levels show strong seasonal variation, populations evolved the ability to tan readily, developing what scientists call facultative pigmentation, moderate baseline pigmentation that can darken significantly in response to increased UV exposure during summer months. Genetic studies examining ancient DNA from archaeological remains have provided remarkable insights into the timing of these changes. For example, analysis of ancient European hunter-gatherers from 7,000 to 8,000 years ago reveals that some still had relatively dark skin despite living in Europe for thousands of years. The widespread shift to very light skin in Europe appears to have accelerated dramatically with the advent of agriculture around 5,000 to 7,000 years ago, possibly linked to dietary changes that affected vitamin D status. Meanwhile, populations that remained in or near the tropics retained dark skin pigmentation. Studies of indigenous populations in Papua New Guinea, Australia, and the Andaman Islands reveal that they share genetic variants for dark skin that are identical by descent from African populations, having been preserved over tens of thousands of years of separation because the selective advantage of dark skin in high UV environments remains strong. The human migration story is also one of repeated contact and interbreeding. As Homo sapiens spread across Eurasia, they encountered other hominin species, Neanderthals in Europe and Western Asia, Denisovans in Central and Eastern Asia, and interbred with them. This genetic exchange transferred some pigmentation-related genes from these archaic humans to modern human populations. Some genetic variants associated with skin color in contemporary Eurasian populations show evidence of being inherited from Neanderthals, who had adapted to Eurasian environments for hundreds of thousands of years before modern humans arrived. These archaic contributions added to the genetic diversity available for natural selection to act upon. This great dispersal, this fanning out of humanity across every inhabitable continent, set the stage for the emergence of the spectacular diversity of skin tones we observe in modern populations. Each population's journey through different environments, each experience of different UV regimes, each series of genetic mutations and selective pressures, contributed threads to the rich tapestry of human diversity. Understanding this migratory history helps us appreciate that skin color is not a fixed racial category but a dynamic, adaptive trait that has responded to environmental conditions throughout our species' history. At the heart of skin color evolution lies a fascinating biological balancing act between two essential nutrients, vitamin D and folate. This elegant explanation for the global pattern of human skin pigmentation, known as the vitamin D folate hypothesis, represents one of the most compelling examples of how natural selection fine-tunes traits to match environmental conditions. Let us delve deeply into this remarkable story of biochemistry, evolution, and adaptation. Vitamin D, often called the sunshine vitamin, is unique among vitamins because our bodies can synthesize it when skin is exposed to ultraviolet radiation. The process begins when UVB photons penetrate the skin and strike a molecule called 7-dehydrocholesterol, which is naturally present in skin cells. The energy from UVB radiation causes a photochemical reaction that converts 7-dehydrocholesterol into pre-vitamin D3. This pre-vitamin then undergoes a temperature-dependent transformation over the course of hours, rearranging itself into vitamin D3, also called cholecalciferol. The newly formed vitamin D3 is released into the bloodstream, where it travels to the liver and then the kidneys, undergoing two hydroxylation reactions that transform it into the biologically active form, 1,25-dihydroxyvitamin D. The importance of vitamin D for human health cannot be overstated. Its most well-known function involves regulating calcium and phosphate metabolism, making it essential for building and maintaining healthy bones and teeth. Without sufficient vitamin D, the body cannot absorb adequate calcium from the diet, leading to soft, weak bones, a condition called rickets in children and osteomalacia in adults. Historical records document epidemics of rickets in industrial-era cities in northern Europe, where smoke and pollution blocked sunlight. Children spent long hours working indoors, and cultural practices including heavy clothing reduced skin's exposure to what limited sunlight was available. But vitamin D's roles extend far beyond bone health. Research over the past few decades has revealed that vitamin D receptors are present in virtually every tissue and cell type in the human body, indicating that this molecule influences a vast array of physiological processes. Vitamin D plays crucial roles in immune system function, helping to activate T cells and regulate inflammatory responses. It influences cardiovascular health, affecting blood pressure regulation and endothelium function. It modulates cell growth and differentiation, with implications for cancer prevention. In pregnant women and developing fetuses, adequate vitamin D is critical for proper fetal skeletal development and may influence birth outcomes. Given its importance and the fact that dietary sources of vitamin D are limited, only fatty fish, fish liver oils, egg yolks, and fortified foods contain meaningful amounts. Humans throughout most of our evolutionary history have relied primarily on cutaneous vitamin D synthesis from sunlight exposure. This dependence creates a fundamental challenge. Melanin, the very pigment that protects skin from UV damage, also blocks the UVB radiation needed for vitamin D synthesis. Studies have quantified this effect with striking clarity. Research shows that people with deeply pigmented skin, Fitzpatrick skin types 5 and 6, require 5 to 10 times more sun exposure than people with lightly pigmented skin, types 1 and 2, to produce the same amount of vitamin D. In one experiment, light-skinned individuals experienced the 50-fold increase in blood vitamin D levels after 8 hours of sun exposure, while dark-skinned individuals exposed to the same sunlight showed no detectable increase. Even with prolonged exposure, dark-skinned individuals achieved only a 30-fold increase, demonstrating that melanin creates a significant barrier to vitamin D synthesis. This presents no problem at equatorial and low latitudes, where UV radiation is intense year-round and even darkly pigmented skin can synthesize adequate vitamin D with routine outdoor activity. But at higher latitudes, particularly above 40 degrees north or south, UVB radiation becomes both weaker and more seasonal. During winter months, the angle of the sun may be so low that virtually no UVB reaches the surface, creating what researchers call the vitamin D winter. a period when cutaneous vitamin D synthesis becomes impossible regardless of skin color or sun exposure. For populations living at these latitudes, dark skin would have posed a serious survival challenge. Vitamin D deficiency during critical periods, pregnancy, lactation, early childhood when bones are growing rapidly, would have reduced reproductive success and childhood survival. Archaeological evidence of rickets in ancient remains from high-latitude populations is relatively rare, suggesting that selection against dark skin in these environments was strong enough to favor lighter pigmentation before populations became established in the far north. This selective pressure for lighter skin to enable vitamin D synthesis represents one side of the evolutionary equation. The other side involves folate, a water-soluble B vitamin, vitamin B9, that is equally crucial for human health and reproduction but has a completely opposite relationship with UV radiation. Folate, particularly its biologically active form called 5-methyltetrahydrofolate, serves essential functions in DNA synthesis, DNA repair, and cell division. This makes it especially critical during periods of rapid cell division, embryonic development, pregnancy, and childhood growth. Folate is required for the proper closure of the neural tube during the first few weeks of pregnancy. Without adequate folate, neural tube defects such as spina bifida can occur. Folate is also essential for spermatogenesis, the production of sperm in males, and for the formation of red blood cells. Folate deficiency leads to megaloblastic anemia, characterized by abnormally large, poorly functioning red blood cells, as well as impaired immune function and increased risk of birth defects. The connection to skin color evolution comes from a crucial discovery. Folate is photosensitive, meaning it is broken down by UV radiation. Research in the late 1970s and 80s demonstrated that exposing human blood plasma to simulated strong sunlight in the laboratory causes a 30 to 50% loss of folate within just 1 hour. Further studies found that light-skinned patients receiving UV light therapy for dermatological conditions developed abnormally low serum folate levels, suggesting that UV photolysis of folate occurs not just in test tubes but in living humans. The mechanism involves both direct photolysis by UV radiation and indirect destruction by reactive oxygen species generated when UVA radiation interacts with photosensitizers present in blood and tissues. The bioactive form of folate, 5-methyltetrahydrofolate, is particularly vulnerable. When UV radiation breaks down this molecule, the damage is irreversible. The folate loses its vitamin activity and cannot be recovered. This discovery provided a compelling explanation for the evolution and maintenance of dark skin in high UV environments. Dark skin, rich in eumelanin, acts as a shield protecting circulating folate from photodegradation. By absorbing and scattering UV radiation before it penetrates to the blood vessels in the dermis, melanin preserves folate levels and prevents the serious consequences of deficiency. The reproductive implications are profound. Folate deficiency during pregnancy increases risks of neural tube defects, premature birth, low birth weight, and maternal complications. In males, folate deficiency impairs sperm production and may reduce fertility. Studies have shown that individuals with lighter skin living in high UV environments, for example, European colonists in Australia, experience higher rates of folate-related problems if they do not adequately protect their skin from sun exposure. Conversely, individuals with dark skin living in low UV environments face different challenges. Their skin effectively protects folate, but they struggle to synthesize sufficient vitamin D. This creates the evolutionary balancing act. Vitamin D requires UV penetration into skin, while folate requires UV protection. Natural selection has solved this problem through skin pigmentation that matches local UV environments. At the equator, where UV is intense year-round, dark skin protects folate while still allowing sufficient vitamin D synthesis. At high latitudes with weak, seasonal UV, light skin maximizes vitamin D production during the limited times when synthesis is possible, while folate protection is less critical because overall UV exposure is lower. Mathematical models based on global UV radiation data have shown remarkably accurate predictions of indigenous skin color distributions. When researchers map UV intensity at different latitudes and seasons, then calculate the optimal skin pigmentation for balancing vitamin D synthesis and folate protection, the predicted skin colors match the observed skin colors of indigenous populations with striking precision. The vitamin D folate hypothesis also explains intermediate pigmentation and tanning ability. Populations living at middle latitudes, approximately between 23 and 46 degrees, experience strong seasonal variation in UV intensity. In these regions, a fixed intermediate pigmentation would be suboptimal year-round, too dark in winter for adequate vitamin D, too light in summer for adequate folate protection. The solution is facultative pigmentation, moderate baseline pigmentation combined with the ability to tan readily in response to increased UV exposure. This allows skin to lighten during winter, maximizing vitamin D synthesis, and darken during summer, protecting folate and preventing sun damage. The delayed tanning response, the mechanism by which skin gradually darkens over days of UV exposure, appears to be an adaptation specifically for these seasonally variable environments. The delay is actually advantageous. In spring, as UV levels begin to rise, the slow development of a tan provides a window for intensive vitamin D synthesis and storage before the full protective tan develops. By the time summer arrives with its intense UV, the tan is fully developed, providing protection during the period of greatest exposure. Competition for folate within the body adds another layer of complexity. Under conditions of high UV stress, the body's folate supplies are simultaneously needed for three major processes: cell division and growth, DNA repair to fix UV-induced damage, and melanin synthesis itself, which requires folate as a cofactor. When UV stress is severe and dietary folate intake is marginal, competition for this limited resource becomes intense. Dark skin helps by reducing UV-induced DNA damage and the resulting need for folate-dependent repairs. Studies examining genetic variants in genes related to folate metabolism have found interesting patterns. Certain variants that affect how efficiently the body uses and recycles folate show different frequencies in populations with different pigmentation levels, suggesting that folate-related genes have co-evolved with skin color genes to optimize folate status in different UV environments. The elegance of the vitamin D folate hypothesis lies in its ability to explain not just the general pattern of human skin color variation, but also many details and exceptions. It explains why women in many populations have slightly lighter skin than men. Women have higher vitamin D requirements during pregnancy and lactation. It explains why populations that adopted agriculture and shifted to grain-based diets, which are low in vitamin D, experienced accelerated evolution toward lighter skin. It explains the persistence of tanning ability in Mediterranean and other middle latitude populations. This is natural selection at its finest, a simple, elegant solution to a complex problem, optimizing multiple competing demands simultaneously through the regulation of a single trait, the amount and type of melanin in skin. The vitamin D folate balance represents a fundamental constraint that has shaped human evolution, health, and diversity in profound ways. The Earth's surface is not uniform in its UV environment. From the scorching equatorial tropics to the dim polar regions, from sea-level coasts to high mountain plateaus, UV radiation varies in intensity, wavelength composition, and seasonal pattern. These geographic variations in UV environment have sculpted equally diverse patterns of human skin pigmentation, creating a living map of evolutionary adaptation that spans the globe. Let us begin our geographic journey at the equator, that imaginary belt around Earth's widest point where the sun passes nearly directly overhead twice each year. Equatorial regions, including parts of East Africa, Central Africa, the Amazon basin, and Indonesia, receive the most intense solar radiation on Earth. Here, the sun's rays travel through the least amount of atmosphere. UV levels remain high throughout the year with minimal seasonal variation. and the combination of intense UVA and UVB creates extraordinary selective pressure for photoprotection. Indigenous populations from these regions display the darkest skin tones found in humans. The Dinka and Nuer peoples of South Sudan, the various ethnic groups of Uganda and Kenya, West African populations from Nigeria and Ghana, and peoples from Papua New Guinea all exhibit deep, rich pigmentation approaching the darkest end of the human spectrum. This pigmentation is not merely aesthetic but functional, a biological necessity for survival and reproductive success in these extreme UV environments. Studies measuring skin reflectance, the amount of light skin reflects at different wavelengths, have found that the darkest skin populations reflect as little as 10 to 20% of visible light, meaning they absorb 80 to 90%. This high absorption extends into the UV range, where melanin's protective effects are most crucial. Genetic analyses reveal that these populations carry multiple genetic variants that maximize eumelanin production, minimize pheomelanin production, and optimize the size, density, and distribution of melanosomes. Interestingly, even within tropical Africa, significant variation exists. Populations in dense rainforest regions, such as the Mbuti and Baka peoples, sometimes have slightly lighter skin than populations in open savanna environments, despite living at similar latitudes. This variation likely reflects differences in actual UV exposure. Rainforest canopies block substantial amounts of UV radiation, and demonstrates how fine-tuned skin color adaptation can be to microenvironmental conditions. Moving away from the equator toward higher latitudes, we observe a general trend toward lighter skin tones, but the pattern is not uniform. The relationship between latitude and skin color follows what scientists call a cline, a gradual change in a trait across a geographic gradient. However, this cline has different slopes and characteristics in different regions of the world. In Africa itself, moving northward from the equator toward the Mediterranean, skin tones gradually lighten. North African populations including Berbers, Egyptians, and other groups along the Mediterranean coast have lighter brown skin compared to sub-Saharan Africans, reflecting reduced UV intensity at these latitudes. However, they maintain substantial pigmentation compared to European populations at similar latitudes, possibly reflecting both the still strong UV exposure in these sunny, arid climates and the more recent timescale of population establishment in these regions. Europe presents a fascinating case study in rapid pigmentation evolution. Archaeological and genetic evidence indicates that early European hunter-gatherers, who arrived in Europe around 45,000 years ago, retained relatively dark skin for thousands of years. Analysis of ancient DNA from remains dating to 7,000 to 8,000 years ago shows that many individuals still had genetic variants associated with darker pigmentation. The widespread evolution of very light skin appears to have occurred relatively recently, accelerating dramatically between 5,000 and 10,000 years ago. Modern indigenous European populations show a striking north-south gradient in skin color. Southern Europeans, Greeks, Southern Italians, Spaniards, Portuguese have olive to moderately brown skin tones and retain strong tanning abilities, reflecting the still substantial UV exposure in Mediterranean climates with long, sunny summers. Moving northward through Central Europe, skin becomes progressively lighter and tanning ability weakens. The northernmost European populations, Scandinavians, particularly Sami people, and northern Russians, display some of the lightest skin tones in the world, with genetic variants that minimize melanin production and maximize the penetration of available UV radiation for vitamin D synthesis. The British Isles present an interesting case. Located at relatively high latitude with famously cloudy weather that further reduces UV exposure, Britain has favored very light pigmentation. Studies have shown that even with maximum sun exposure during British summer, deeply pigmented individuals struggle to synthesize adequate vitamin D. The evolution of light skin in these populations was likely driven by intense selective pressure, as vitamin D deficiency would have had severe consequences for bone development and overall health. Northern Asia tells a similar but genetically distinct story. Indigenous populations from Siberia, northern China, Mongolia, and Japan show a parallel evolution toward lighter skin at high latitudes, but this lightening occurred through different genetic changes than in Europeans. The key mutations affecting skin color in East Asian populations occurred in genes like KITLG and OCA2 rather than the SLC24A5 and SLC45A2 variants that swept through Europe. This independent evolution of light skin through different genetic pathways provides powerful evidence for convergent evolution. Natural selection arriving at similar solutions through different genetic routes. Interestingly, East Asian populations tend to have a slightly yellower undertone to their skin compared to Europeans at similar latitudes. This reflects differences in the proportion of different melanin types and possibly differences in other pigments present in skin, such as carotene. These subtle differences in skin tone, while minor, remind us that skin color is not simply a one-dimensional dark to light scale but a multidimensional spectrum influenced by multiple biological factors. South Asia presents perhaps the most complex skin color geography in the world. The Indian subcontinent spans from near equatorial latitudes in the south to subtropical and temperate latitudes in the north, and correspondingly, skin tones show enormous variation. Southern Indian populations tend to have darker brown skin, while northern populations show lighter brown to olive tones. However, this pattern is complicated by thousands of years of migration, admixture, and social stratification. Genetic studies have revealed that South Asian populations carry a mix of ancestry from multiple sources. Ancient hunter-gatherers who inhabited the region for tens of thousands of years, farmers who migrated from the Middle East bringing agricultural technologies, and pastoralists who arrived from Central Asian steppes. Each group brought different genetic variants affecting pigmentation, and the complex patterns of intermixing and isolation among different communities have created extraordinary diversity. Some South Asian populations carry the SLC24A5 variant common in Europeans, introduced through ancient gene flow from Western Eurasia, while others lack this variant entirely, relying on different genetic variants to achieve lighter pigmentation at northern latitudes. The Americas present their own intriguing patterns. The first peoples to colonize the Americas crossed from Asia via the Bering Land Bridge probably between 15,000 and 20,000 years ago, though possibly earlier. These founding populations brought with them the genetic variant for lighter skin that had evolved in their East Asian ancestral populations. As they dispersed southward through the Americas, different populations encountered different UV environments. Indigenous populations from near equatorial regions in the Amazon basin, Central America, and the Andean highlands developed moderately brown skin, lighter than African or Melanesian populations at similar latitudes. This has puzzled researchers who initially expected tropical American populations to evolve very dark skin given the high UV exposure. Several factors may explain this pattern. First, the colonization of the Americas is relatively recent on evolutionary timescales, possibly not providing enough time for new mutations producing very dark skin to arise and spread. Second, clothing and shelter technologies, well developed in Asian populations before migration to the Americas, may have buffered UV exposure enough to reduce selective pressure for the darkest pigmentation. Third, high-altitude environments. Many tropical American populations live at elevations where UV is intense but temperatures are cool, may create unique selective pressures. The Inuit peoples of the Arctic present an important exception to the general latitude pigmentation relationship. Despite living at extreme northern latitudes where one might expect very light skin, traditional Inuit populations maintain moderately brown skin, darker than Europeans at the same latitude. This apparent anomaly has several explanations. Inuit traditional diet, rich in fatty fish and marine mammals, provides abundant dietary vitamin D, relaxing the selective pressure for light skin to maximize vitamin D synthesis. Additionally, the highly reflective snow and ice of Arctic environments create intense UV exposure from reflected radiation. particularly in spring and summer, maintaining selective pressure for photoprotection. Finally, the relatively recent arrival of Inuit ancestors in the Arctic, within the past 5,000 years, may not have provided sufficient time for extreme depigmentation to evolve. Australia and Melanesia showcase yet another pattern. Indigenous Australians, who have inhabited the continent for at least 65,000 years, maintain very dark skin despite living as far as 40 degrees south latitude in southern Australia, latitudes where European Australians and other populations have very light skin. Similarly, Melanesian populations inhabiting tropical Pacific islands maintain extremely dark pigmentation. Genetic studies reveal that these populations inherited variants for dark skin directly from their African ancestors, preserving them because the strong UV exposure in these regions. Australia is notorious for having the highest UV levels and skin cancer rates in the world, maintains selective pressure for photoprotection despite the higher latitude. The global pattern of indigenous skin color, when mapped against UV radiation data, creates a remarkably coherent picture. If you were to overlay a map of human skin pigmentation onto a map of annual UV exposure, you would observe a stunning correlation. The darkest skin tones cluster in regions receiving the most intense UV radiation, the equatorial belt stretching across Africa, Southeast Asia, and Oceania. Moving toward the poles in both hemispheres, skin tones lighten progressively, reaching their palest extremes in the far northern reaches of Europe and Asia. This geographic distribution is not random or arbitrary but represents hundreds of thousands of years of fine-tuned adaptation to local UV environments, with each population's pigmentation optimized for balancing vitamin D synthesis against full light protection in their ancestral homeland. Having explored the geographic patterns of human skin color across the globe, we now turn our attention inward, to the molecular machinery that produces this magnificent diversity. The story of how skin color is inherited is a tale of multiple genes working in concert, each contributing its own influence to create the final phenotype, the observable skin color we see. Skin color is what geneticists call a polygenic trait, meaning it is influenced by many genes rather than a single gene. This polygenic nature explains why skin color shows continuous variation rather than discrete categories, why children's skin tones often fall somewhere between their parents' tones, and why even full siblings can display noticeably different pigmentation. Current research has identified more than 150 genetic loci, positions in the genome, associated with skin pigmentation, though a smaller number of genes account for most of the variation between populations. Let us explore the key players in this genetic orchestra, beginning with MC1R, the melanocortin-1 receptor gene. This gene encodes a protein receptor located on the surface of melanocytes. When a signaling molecule called alpha-melanocyte-stimulating hormone binds to this receptor, it triggers a cascade of biochemical reactions that ultimately increase eumelanin production. Think of MC1R as a switch that, when activated, turns on the production of dark melanin. Functional variants of MC1R promote high eumelanin production and dark pigmentation. However, mutations that disrupt MC1R's function interfere with this signaling pathway, shifting melanin production away from protective eumelanin toward pheomelanin, the red-yellow pigment that offers less UV protection. Studies of MC1R variation across populations reveal fascinating patterns. In African populations, particularly those from equatorial regions, MC1R shows remarkably low diversity. Nearly all individuals carry fully functional versions of the gene, reflecting strong negative selection, natural selection that removes harmful variants from the population. Any mutation that reduced MC1R function and decreased eumelanin production would have been strongly selected against because lighter skin would have been severely disadvantageous in high UV environments. The low diversity of MC1R in African populations represents one of the clearest examples of purifying selection in the human genome. In contrast, European populations display extraordinary MC1R diversity. with numerous variants that partially or completely disrupt the receptor's function. These variants are associated with red hair, fair skin, freckles, and increased susceptibility to sunburn and skin cancer. The most common of these variants shows frequencies of 10 to 20% in some European populations. The high frequency of loss of function MC1R variant in Europe indicates that once human populations move to low UV environments, the strong selective pressure maintaining MC1R function was relaxed. Without intense UV radiation creating health consequences for reduced eumelanin production, genetic variants that would have been harmful in Africa could persist and even increase in frequency in European populations. The fact that MC1R variants remain polymorphic rather than becoming fixed suggests that there may still be some selective cost to completely non-functional MC1R, possibly related to increased skin cancer risk. But this cost is evidently much lower in low UV environments. Another critical gene is SLC24A5, which encodes a protein called NCKX5 that functions as a transporter on the membrane of melanosomes. This protein plays a crucial role in regulating the ionic environment inside melanosomes, influencing the efficiency of melanin synthesis. A single nucleotide polymorphism in SLC24A5, a change at one specific position in the DNA sequence, results in an amino acid substitution that dramatically affects pigmentation. The ancestral version of this gene, found at nearly 100% frequency in African populations, East Asian populations, and Native American populations, is associated with dark skin. The derived variant, which arose through mutation, substantially reduces melanin production and is associated with lighter skin. What makes SLC24A5 particularly fascinating is its distribution. The lightning variant is found at frequencies approaching 100% in European populations, meaning nearly every European carries 2 copies of this variant. However, this same variant is virtually absent in East Asian populations, who have evolved light skin through entirely different genetic mechanisms. This pattern provides powerful evidence for convergent evolution. Europeans and East Asians independently evolved lighter skin to adapt to similar low UV environments, but they did so through different genetic pathways. The story of how the SLC24A5 lightning variant was discovered adds an interesting twist. Scientists studying zebrafish identified a mutation in the fish version of this gene that caused golden coloration. When they investigated the human equivalent, they discovered the variant that accounts for a large portion of pigmentation difference between Africans and Europeans. Studies estimate that this single genetic variant accounts for approximately 25 to 38% of the skin pigmentation difference between West Africans and Europeans, an extraordinarily large effect for a single gene. Genetic analyses examining the pattern of DNA variation surrounding SLC24A5 in European populations reveal a classic signature of positive selection. The region of the genome containing this gene shows dramatically reduced genetic diversity in Europeans compared to other populations, with all the copies of the lightning variant appearing nearly identical. This pattern indicates that the variant arose relatively recently and spread rapidly through European populations because it conferred a strong selective advantage. Estimates suggest this sweep occurred within the past 11,000 to 19,000 years, relatively recently in human evolutionary terms, possibly accelerating with the adoption of agriculture. SLC45A2, formerly known as MATP, is another gene with major effects on pigmentation. Like SLC24A5, it encodes a transporter protein located on melanosome membranes and influences melanosome pH and melanin synthesis efficiency. A key variant in SLC45A2 that substantially reduces pigmentation shows similar patterns to SLC24A5. It reaches high frequencies in European populations but is absent or rare in East Asian and African populations. Studies suggest this variant also underwent positive selection in Europeans, with the sweep occurring within a similar time frame. Together, Variants in SLC24A5 and SLC4A5A2 account for a substantial proportion of the light skin phenotype in Europeans. The TWAR gene encodes tyrosinase, the crucial enzyme that catalyzes the first rate-limiting step in melanin synthesis, the conversion of the amino acid tyrosine into dopa and then dopaquinone. Without functional tyrosinase, no melanin can be produced at all. Individuals who inherit two non-functional copies of TWAR have oculocutaneous albinism type 1, characterized by a complete absence of melanin in skin, hair, and eyes. Less severe mutations in TWAR result in partial reductions in melanin production and contribute to natural variation in pigmentation. Several TWAR variants associated with lighter skin show increased frequencies in European populations and show evidence of positive selection. OCA2, which stands for oculocutaneous albinism type 2, encodes a protein that regulates melanosin pH and appears to influence the trafficking of tyrosinase and other melanogenic proteins within melanocytes. Complete loss of OCA2 function results in albinism, but less severe variants contribute to normal pigmentation variation. Interestingly, genetic variants near the OCA2 gene, in a neighboring regulatory region called HERT2, are the primary genetic determinants of blue versus brown eye color in Europeans. The same variants that produce blue eyes also contribute to lighter skin, demonstrating how genetic variants can have pleiotropic effects, influencing multiple traits simultaneously. In East Asian populations, the genetic basis of light skin differs markedly from Europeans. While some genes like OCA2 and KITLG show evidence of selection in both Europeans and East Asians, the majority of genes under selection differ between the two populations. East Asian-specific targets of selection include genes like DCT, which encodes dopachrome tautomerase, an enzyme involved in melanin synthesis, ADAM17 and ADAM20, which code for proteases involved in cellular signaling, BNC2, a transcription factor regulating melanocyte gene expression, LYST, involved in organelle trafficking, and TYPR1, related to pigmentation pathways. The different genes under selection in East Asians versus Europeans demonstrate that multiple genetic pathways can be modulated to achieve similar phenotypic outcomes. Lighter skin adapted to low UV environments. Studies analyzing ancient DNA from archaeological remains have provided direct evidence of the timing of pigmentation evolution in Europe. DNA extracted from hunter-gatherers who lived in Europe 7,000 to 8,000 years ago reveals that many individuals carried ancestral variants for darker pigmentation at key genes like SLC24A5 and SLC45A2, despite living in Europe for thousands of years. This suggests that the widespread shift toward very light skin in Europe occurred relatively recently and may have accelerated with the arrival of farming populations from the Middle East around 8,000 years ago, who brought lightening variants with them and had mixed with European hunter-gatherers. The mechanism by which skin color is inherited follows basic principles of genetic inheritance, but with added complexity due to the involvement of multiple genes. Each parent contributes one copy of each gene to their offspring, who thus inherits two copies total, one from each parent. For any given pigmentation gene, an individual might inherit two copies of a lightening variant, two copies of a darkening variant, or one of each. The overall skin color results from the combined effects of variants at all the relevant genes. Because multiple genes contribute to skin color, each adding or subtracting a small amount from the total melanin production, the range of possible phenotypes is large and continuously distributed. Two parents with intermediate skin tones could produce children ranging from lighter than either parent to darker than either parent, depending on which combination of variants each child happens to inherit. This explains the wide variation sometimes observed among siblings. The inheritance pattern also explains why skin color often appears to blend across generations. When individuals from populations with very different ancestral pigmentation have children together, the offspring typically display intermediate pigmentation, though with substantial variation. These children inherit a mixture of lightening and darkening variants from their two parents, and the overall effect typically falls somewhere in between. Modern genetic research using genome-wide association studies, which scan the entire genome looking for genetic variants associated with particular traits, continues to identify new genes and variants influencing pigmentation. As studies expand to include more diverse populations beyond Europeans, researchers are discovering additional variants that contribute to pigmentation variation in African, South Asian, and other populations. These studies reveal that even within Africa, despite the overall maintenance of dark pigmentation, there is substantial genetic variation contributing to the range of skin tones observed across the continent. Some of this within African variation involves the same genes that vary between populations. For instance, in southern African populations like the Khoisan, who have relatively lighter brown skin compared to equatorial Africans, researchers have found higher frequencies of variants in genes like SLC24A5, SLC45A2, and KITLG that are typically associated with lighter pigmentation. This appears to represent convergent evolution. Just as populations moving to high latitudes evolved lighter skin, populations in southern Africa, which receives less UV than equatorial regions, have evolved moderately reduced pigmentation through selection on similar genes. The complexity of skin color genetics has important implications for understanding human diversity. Because pigmentation is controlled by many genes with varying effect sizes, because different populations have evolved light skin through different genetic pathways, and because there is substantial genetic variation within populations, skin color does not serve as a reliable marker for overall genetic ancestry or relatedness. Two individuals with similar skin color might have very different overall genetic backgrounds, and two individuals with different skin colors might be genetically similar across the rest of their genomes. Furthermore, the genes that control skin pigmentation represent a tiny fraction of the total human genome, just a few dozen genes among more than 20,000 protein-coding genes. The genetic variants that differ in frequency between populations and contribute to pigmentation differences represent an even smaller fraction of human genetic variation. The vast majority of human genetic variation exists within populations rather than between them, and the genes controlling skin color are among the relatively few regions of the genome showing strong between-population differences due to local environmental adaptation. Understanding the genetic basis of skin color thus provides insight not just into how this particular trait evolved, but into how evolution works more broadly. It demonstrates how natural selection can act powerfully on specific genes responding to environmental pressures while leaving most of the genome relatively unaffected. It shows how similar adaptive solutions can evolve independently through different genetic mechanisms. And it reveals how complex traits can emerge from the combined action of many genes, each contributing incrementally to the final phenotype. While latitude and its associated UV radiation intensity represent the dominant environmental driver of skin color evolution, other environmental factors have also played important roles in shaping human pigmentation patterns. These additional factors create exceptions to the general latitude pigmentation relationship and remind us that evolution responds to the totality of environmental conditions, not just a single variable. Altitude represents one such factor. As elevation increases, UV radiation intensity increases dramatically because there is less atmosphere above to absorb and scatter the radiation. For every 1,000 meters of elevation gain, UV intensity increases by approximately 10 to 12%. This means that populations living at high altitude experience UV exposure comparable to or even exceeding that at sea level much closer to the equator. The thin atmosphere at high elevations allows more UV radiation to reach the surface, creating intense selective pressure for photoprotection even at latitudes where lowland populations have relatively light skin. The effect of altitude on pigmentation can be observed in several regions. In the Andes Mountains of South America, Indigenous populations living at elevations of 3,000 to 5,000 meters maintain moderately brown skin despite living at middle latitudes where sea level populations in other regions have much lighter skin. The Tibetan Plateau, averaging over 4,000 meters in elevation, receives intense UV exposure despite its northern latitude. Tibetan populations, while not as darkly pigmented as equatorial Africans, maintain substantially more pigmentation than Chinese populations living at similar latitudes in low elevation regions. Studies have identified genetic variants unique to Tibetan populations that may represent adaptations not just to the low oxygen levels at high altitude but also to the intense UV environment. The relationship between altitude and pigmentation demonstrates that UV exposure, the actual amount of radiation reaching skin, matters more for adaptive pigmentation than latitude alone. Bodies of water, vegetation coverage, atmospheric conditions, and topography all modulate UV exposure in ways that can override the general latitude effect. Populations living in cloudy, rainy regions receive less UV exposure than those at similar latitudes in sunny, arid climates, and pigmentation patterns reflect these differences. Diet has also influenced skin color evolution in complex ways. As mentioned earlier, vitamin D can be obtained not just from sunlight-driven synthesis but also from food sources. Throughout most of human evolution, dietary vitamin D was scarce. Natural food sources of vitamin D are limited primarily to fatty fish, fish liver oils, egg yolks from certain birds, and some mushrooms. Most traditional human diets contain very little vitamin D, making cutaneous synthesis from sunlight the primary source. However, populations with access to rich dietary vitamin D sources faced somewhat reduced selective pressure for light skin to maximize vitamin D synthesis. The clearest example comes from Arctic populations whose traditional diets consisted largely of marine mammals and fish, providing abundant dietary vitamin D. Inuit peoples, despite living at extreme northern latitudes with long winter darkness, maintain moderately brown skin that would seem maladaptive for vitamin D synthesis. The explanation lies in their diet. Consuming seal, whale, and fish provides so much vitamin D that cutaneous synthesis becomes less critical, relaxing the selective pressure for depigmentation. Studies comparing vitamin D status across populations support this interpretation. Traditional Inuit populations consuming their ancestral marine mammal-based diet maintain adequate vitamin D status despite dark skin and limited UV exposure. In contrast, when Inuit populations adopt Western diets low in fatty fish and marine mammals, they experience high rates of vitamin D deficiency unless they take supplements. This demonstrates that the moderate pigmentation of Inuit populations was maintained, at least in part, because dietary vitamin D buffered them against deficiency despite living in low UV environments. The shift to agriculture approximately 10,000 years ago in various regions may have influenced skin pigmentation evolution. Agricultural diets based on grains and cereals contain very little vitamin D, creating nutritional stress that would have increased selective pressure for light skin to maximize vitamin D synthesis from the limited sun exposure available to farming populations who spent more time indoors compared to hunter-gatherers. Some researchers hypothesize that the apparent acceleration of skin lightening in European populations around 5,000 to 10,000 years ago may be linked to the spread of agriculture and the resulting dietary changes. Cultural practices involving clothing, shelter, and daily activity patterns have also modulated selective pressures on pigmentation. Humans are unique among primates in wearing clothing, and clothing coverage significantly reduces UV exposure to skin. Populations that adopted clothing early in their history would have experienced reduced UV exposure compared to populations with less clothing coverage at similar latitudes. However, clothing also reduces vitamin D synthesis by blocking UV radiation, potentially increasing selective pressure for lighter skin to maximize synthesis from whatever skin remains exposed. The specific styles of clothing and their coverage levels would have mattered. Desert populations often wear extensive clothing coverage for protection from heat and sun, reducing direct skin UV exposure despite living in high UV environments. Some researchers have suggested that certain populations in hot, arid regions may have evolved moderately lighter skin than their UV environment alone would predict because extensive clothing coverage reduced the selective pressure for maximal pigmentation. Behavioral practices around sun exposure would have influenced selection on pigmentation. Populations that developed cultural practices of avoiding midday sun or seeking shade during the hottest hours would have experienced reduced UV exposure compared to populations without such practices. Activities that required extended outdoor exposure during peak UV hours, such as certain forms of agriculture, animal herding, or fishing, would have increased UV exposure and maintained stronger selection for protective pigmentation. Indoor lifestyle shifts in recent centuries have dramatically altered UV exposure patterns for many populations, but on too short a timescale for significant genetic evolutionary change. Modern humans in industrialized societies spend the vast majority of their time indoors, often receiving only a fraction of the sun exposure their ancestors experienced. This lifestyle change has created widespread vitamin D deficiency in many populations, particularly affecting individuals with darker skin living in low UV regions who now receive insufficient UV exposure for adequate vitamin D synthesis. This represents a mismatch between our evolved physiology and modern environmental conditions. Our skin color adapted to our ancestors' UV exposure levels, but we now live very differently. Seasonal migration patterns in some traditional societies created complex UV exposure scenarios. Populations that practiced transhumance, seasonal movement to different elevations or latitudes following herds or agricultural cycles, experienced varying UV intensities throughout the year. These populations might have benefited from moderate pigmentation with strong tanning ability, allowing for seasonal adjustment of photoprotection and vitamin D synthesis. Cloud cover and atmospheric conditions significantly affect ground-level UV intensity. Regions with persistent cloud cover or high air pollution receive substantially less UV than clear sky regions at similar latitudes. Historical records from industrial-era European cities document epidemics of rickets among children, caused by vitamin D deficiency resulting from the combination of indoor living, heavy clothing, and air pollution from coal burning that blocked UV radiation. While this time scale is too short for genetic evolutionary response, it illustrates how atmospheric conditions modulate UV exposure and create health consequences related to vitamin D. Proximity to water can also influence UV exposure through reflection. Water surfaces reflect substantial amounts of UV radiation, increasing total exposure for people living and working near water. This effect is particularly pronounced near snow and ice, which reflect up to 90% of UV radiation. Arctic and Antarctic environments, despite low direct UV due to high latitude and sun angle, actually deliver intense UV exposure due to reflection from snow and ice. This reflected UV maintains selective pressure for pigmentation in high-latitude regions with extensive snow cover. The interplay between these various environmental factors, altitude, diet, clothing, cultural practices, lifestyle, and atmospheric conditions, creates the complex pattern of human pigmentation observed worldwide. While latitude and its associated UV intensity represent the primary driver of pigmentation evolution, these additional factors fine-tune the pattern, creating local adaptations and explaining apparent exceptions to the general latitude pigmentation relationship. Understanding these environmental complexities reminds us that evolution does not optimize traits for a single environmental variable, but for the complex, multifaceted conditions organisms actually experience. Human skin color represents a beautiful example of multifactorial adaptation, shaped by the intricate interaction of physical environment, nutritional ecology, and cultural practices, all filtered through the constraint of maintaining the delicate vitamin D folate balance essential for health and reproduction. As we emerge from the deep evolutionary past into the present day, We must confront the modern implications of human skin color diversity and address the persistent misunderstandings that continue to cause harm in contemporary societies. The scientific understanding of skin color as an adaptive trait evolved in response to environmental UV radiation stands in stark contrast to the social constructions and prejudices that have been built around superficial physical differences. First and foremost, we must acknowledge the health implications of skin color in modern environments. The mismatch between ancestral UV environments and current living conditions creates health challenges for many populations. Individuals with deeply pigmented skin living in low UV high-latitude regions face significant risk of vitamin D deficiency. The melanin that protected their ancestors from intense tropical UV now inhibits the synthesis of adequate vitamin D from the weak, seasonal sunlight available at northern latitudes. Research has documented widespread vitamin D deficiency among dark-skinned populations in northern regions. Studies in the United Kingdom found that individuals of African and South Asian descent living in Britain have dramatically higher rates of vitamin D deficiency compared to the white British population. One study found that vitamin D deficiency, defined as serum levels below 25 nanomoles per liter, affected 61% of South Asian women and 58% of African Caribbean women, compared to just 7% of white women. Even more concerning, severe deficiency below 12.5 nanomoles per liter affected 19% of South Asian women and 14% of African Caribbean women, compared to less than 1% of white women. The health consequences of this deficiency are serious. Rickets, the softening and weakening of bones in children due to vitamin D deficiency, has re-emerged as a public health concern in the UK and other northern countries, primarily affecting children of African, South Asian, and Middle Eastern descent. In adults, vitamin D deficiency contributes to osteomalacia, characterized by soft, weak bones and chronic bone pain. Beyond skeletal health, vitamin D deficiency has been associated with increased risk of respiratory infections, autoimmune conditions, cardiovascular disease, certain cancers, and depression. Pregnant women with vitamin D deficiency face increased risks of pregnancy complications, and their infants may have impaired bone development. These health disparities represent a direct consequence of evolutionary adaptation meeting modern circumstances. Dark skin, perfectly adapted for equatorial UV environments, becomes a liability in low UV regions, particularly when combined with modern indoor lifestyles that further reduce sun exposure. Public health responses have included recommendations for vitamin D supplementation for at-risk populations. Health authorities in the UK, for example, recommend that everyone consider taking vitamin D supplements during winter months, with particular emphasis on individuals with dark skin who face higher deficiency risk year-round. Conversely, individuals with very light skin, particularly those with MC1 or variants producing red hair and fair, freckling skin, face elevated skin cancer risk. The same light pigmentation that facilitates vitamin D synthesis in low UV environments offers inadequate protection in high UV regions. Australia and New Zealand, with predominantly European descent populations living under intense UV, have the highest skin cancer rates in the world. In Australia, approximately 2 out of 3 people will be diagnosed with skin cancer by age 70. Melanoma, the deadliest form of skin cancer, causes over 1,500 deaths annually in Australia. The risk is particularly acute for individuals with very fair skin who sunburn easily, those with many moles, those with red hair, and those with a family history of skin cancer. Studies have shown that individuals with certain MC1R variants have 2 to 4 times higher melanoma risk compared to those with fully functional MC1R, independent of sun exposure. This elevated risk reflects reduced photoprotection. The pheomelanin produced when MC1R is non-functional provides minimal UV protection and may even contribute to oxidative damage when exposed to UV radiation. Public health campaigns in high UV regions emphasize sun protection. Seeking shade during peak UV hours, wearing protective clothing including hats and long sleeves, and applying broad-spectrum sunscreen regularly. These recommendations are particularly important for light-skinned individuals and children, as childhood sunburns significantly increase lifetime melanoma risk. The Australian Slip, Slop, Slap campaign, slip on a shirt, slop on sunscreen, slap on a hat, represents one of the most successful public health messaging efforts targeting skin cancer prevention. An interesting complexity arises from the relationship between sun exposure, vitamin D, and melanoma prognosis. Research has found that melanoma patients with higher vitamin D levels at diagnosis tend to have better outcomes, including thinner tumors and improved survival. This creates a challenging paradox. Sun exposure causes melanoma but also produces vitamin D that may improve melanoma outcomes. The current medical consensus emphasizes that for individuals with melanoma or at high melanoma risk, sun protection should be maintained and vitamin D adequacy should be achieved through diet and supplementation rather than sun exposure. Beyond these direct health implications, we must address the profound social and historical misuse of skin color to justify racism, discrimination, and oppression. For centuries, superficial physical differences including skin color have been misused to construct false racial hierarchies, justify slavery and colonialism, and rationalize discrimination and violence. These racist ideologies have caused immeasurable harm and continue to affect societies worldwide. Modern scientific understanding of skin color evolution thoroughly debunks racist ideology. First, skin color is a single trait controlled by a relatively small number of genes, representing a tiny fraction of human genetic diversity. The genes that vary in frequency between populations and affect pigmentation are exceptional precisely because they show strong between-population differences due to local environmental adaptation. The vast majority of human genetic variation, approximately 85 to 90%, exists within populations rather than between them. Two individuals from the same population are almost as genetically different from each other as two individuals from different populations. Second, the racial categories historically constructed based on skin color do not correspond to meaningful biological divisions. Genetic studies have repeatedly demonstrated that human genetic variation is continuous and clinal, gradually changing across geography, rather than falling into discrete categories. The boundaries of socially constructed racial groups are arbitrary and inconsistent, varying across cultures and historical periods. Someone classified as black in the United States might be classified differently in Brazil, South Africa, or other societies with different racial classification systems. Third, the genetic variants producing similar skin colors in different populations often differ, reflecting convergent evolution. Europeans and East Asians independently evolved lighter skin through different genetic changes. Someone with light skin from Europe and someone with light skin from East Asia share similar pigmentation despite having different genetic variants producing that phenotype. This demonstrates that similar appearance does not imply shared ancestry or genetic similarity across the genome. Fourth, the traits that vary visibly between populations, skin color, hair texture, facial features, body proportions, represent adaptations to climate and other local environmental conditions. These traits evolved relatively recently in human history and affect only a tiny fraction of human biology. They provide no information about intelligence, personality, behavior, or any other complex traits that some racist ideologies have falsely associated with race. The scientific consensus, supported by overwhelming evidence, is that there are no biologically meaningful racial categories among humans and no genetic basis for racial hierarchies. Understanding skin color evolution helps combat racism by revealing that pigmentation differences are precisely what evolutionary biology predicts: adaptive responses to different UV environments. There is nothing more profound or meaningful about differences in melanin production than there is about differences in altitude adaptation or lactose tolerance or any other geographically variable trait shaped by natural selection. Skin color carries no information about cognitive abilities, moral character, cultural achievements, or any other traits used to justify discrimination. The persistence of racism despite scientific evidence demonstrates that racism is a social and political phenomenon, not a biological reality. Racist ideologies were constructed to justify economic exploitation, colonial conquest, and social hierarchies. And they persist because they serve certain social and political functions, not because they reflect biological truth. Education about the evolutionary biology of skin color can help counter racist myths, but dismantling racism requires addressing its social, economic, and political dimensions, not just providing scientific education. A related misunderstanding involves the concept of race itself. Many people believe that races are natural, biological divisions among humans. Modern genetics has thoroughly refuted this notion. While human populations show genetic variation related to geographic ancestry, populations that have been geographically separated for long periods have somewhat different frequencies of genetic variance. This variation is continuous and clinal rather than falling into discrete categories. The idea of distinct races is a social construction imposed on continuous biological variation. Genetic ancestry testing has become popular in recent years, with companies offering to tell customers what percentage of various ancestries they carry. While these tests can provide interesting information about geographic origins of ancestors, they can also perpetuate misunderstandings about race and biology. The ancestry categories used by testing companies are arbitrary, and the percentages provided are estimates with substantial uncertainty. More importantly, genetic ancestry does not determine identity, culture, or social experience. Someone who learns they have ancestry from multiple continents still experiences the world based on how society perceives and treats them, not based on genetic percentages. The modern understanding of skin color also has implications for medical practice. Historically, medical research and drug testing have been conducted primarily on populations of European descent, leading to gaps in knowledge about how conditions present and respond to treatment in individuals with different ancestral backgrounds. There is growing recognition of the need for more diverse inclusion in medical research. However, this need for better representation must be balanced against the danger of reifying race as a biological category in medicine. Skin color and geographic ancestry can correlate with certain health risks. For example, sickle cell trait is more common in individuals with ancestry from regions where malaria is endemic, and cystic fibrosis is more common in individuals of European descent. But these correlations reflect population history and environmental adaptation, not essential racial differences. Treating patients based on crude racial categories rather than individual assessment can lead to suboptimal care. A more nuanced, individualized approach to medicine recognizes that human genetic variation is continuous and considers individual genetic background, family history, lifestyle, and environment rather than relying on racial categories. This approach maintains the benefits of understanding population differences in disease risk while avoiding the harmful oversimplifications of race-based medicine. The evolution of skin color also provides a powerful example for education about evolution in general. The global pattern of pigmentation adapting to UV radiation is one of the clearest, most compelling demonstrations of natural selection in action. Students can see how environmental pressures select for traits that enhance survival and reproduction, how similar challenges in different populations lead to convergent evolution, and how traits that appear fixed and essential are actually recent, reversible adaptations to local conditions. Teaching about skin color evolution can help students understand that human diversity is a product of adaptation to varied environments, not evidence of fundamental differences between groups. It demonstrates that evolution is ongoing, that humans are still evolving, and that our bodies bear the marks of our ancestral environments even when we now live in very different circumstances. It illustrates how scientific understanding can challenge and refute harmful ideologies. In summary, the modern implications of human skin color diversity are multifaceted. They include direct health consequences of mismatches between evolved pigmentation and current UV environments, requiring public health interventions around vitamin D supplementation and sun protection. They include the imperative to understand and combat racist ideologies that have misused superficial physical differences to justify oppression. They include opportunities for education about evolution, adaptation, and the true nature of human biological diversity. And they include challenges for medicine to appropriately account for human variation while avoiding the reification of false racial categories. Understanding skin color as an evolved, adaptive trait responding to environmental UV radiation provides crucial context for navigating these modern complexities. The story of human skin color evolution is not finished. Evolution continues in the present day, and new forces are shaping the trajectory of human pigmentation. Understanding these ongoing processes and considering future scenarios provides insight into human adaptability and the dynamic nature of our species. Human migration and gene flow are reshaping global patterns of genetic variation, including pigmentation-related genes. Throughout most of human history, populations remained relatively isolated geographically, with limited migration and admixture between distant groups. This isolation allowed populations to develop distinct genetic profiles adapted to their local environments. However, over the past several centuries, and particularly in recent decades, human migration has accelerated dramatically, bringing together individuals from populations that diverged tens of thousands of years ago. The genetic consequences of this increased migration and admixture are profound. In many regions, populations that were historically distinct are now mixing extensively. In the Americas, populations with ancestry from Europe, Africa, and indigenous America have been mixing for 500 years, creating admixed populations with complex genetic ancestry. Recent migration has increased mixing among populations worldwide, from South Asians in Britain to Africans in Europe to Latin Americans in the United States. This admixture creates populations with intermediate pigmentation, as individuals inherit combinations of lightening and darkening genetic variants from ancestrally distinct parents. Studies of admixed populations provide valuable information about the genetic architecture of pigmentation because the trait varies within populations due to varying proportions of ancestry from groups with different pigmentation. Research in African American and Hispanic American populations has helped identify and confirm several genes affecting skin color by comparing pigmentation to genetic ancestry at different regions of the genome. Looking forward, continued admixture will likely reduce the correlation between pigmentation and geographic origin. If current migration patterns continue, future human populations may show more continuous variation in skin color that is less strongly structured by geography. However, this process will take many generations to substantially alter global patterns, and new migration restrictions or population movements could change trajectories. Another ongoing evolutionary force is sexual selection on pigmentation. In many cultures, skin color affects mate preferences with implications for evolutionary change. Cultural ideals about attractiveness vary across societies, with some favoring lighter skin and others valuing darker skin. In societies where lighter skin is considered more attractive, individuals with lighter pigmentation may have reproductive advantages through increased mating success, potentially driving evolution toward lighter pigmentation over many generations. Conversely, if cultural preferences shift toward valuing darker skin, this could drive evolution in the opposite direction. However, the strength of sexual selection on pigmentation is likely much weaker than the historical selective pressures from UV radiation and vitamin D folate balance. Cultural preferences for skin color are also variable, change over time, and are influenced by complex social factors including historical legacies of colonialism and racism. Whether sexual selection will produce significant evolutionary change in human pigmentation remains uncertain. Modern medicine and public health interventions have relaxed natural selection on pigmentation in some ways. Vitamin D supplementation and fortification of foods with vitamin D reduced the health consequences of inadequate vitamin D synthesis. potentially relaxing selective pressure for light skin in low UV environments. Similarly, sunscreens, protective clothing, and skin cancer surveillance reduce the health consequences of inadequate photoprotection, potentially relaxing selection for dark skin in high UV environments. However, these interventions are unevenly distributed globally. Many populations lack access to vitamin D supplementation, fortified foods, sunscreens, and medical care. In these populations, natural selection on pigmentation related to vitamin D and UV protection likely continues. Even in populations with access to medical interventions, compliance is imperfect, and some health effects of pigmentation may persist. Climate change introduces new considerations for future skin color evolution. As global temperatures rise and weather patterns shift, UV exposure patterns are changing in complex ways. Ozone layer recovery may reduce UV exposure in some regions, while changes in cloud cover, air pollution levels, and snow cover may increase or decrease UV in others. Sea level rise, desertification, and other climate impacts may drive human migration to regions with different UV environments, creating new selective pressures. Lifestyle changes, particularly the transition to indoor living in industrialized societies, have dramatically altered UV exposure for much of humanity. Modern humans in developed nations spend the vast majority of time indoors, exposed to artificial lighting that provides no UV radiation. This lifestyle shift has created widespread vitamin D deficiency and may be creating new selective pressures, though the timescale of evolution means that significant genetic change would require thousands of years. If humanity continues its current trajectory toward indoor living, with UV exposure declining to very low levels regardless of latitude, selection on pigmentation might shift. In principle, if UV exposure becomes minimal worldwide due to lifestyle, the protective function of dark skin becomes less important, potentially allowing pigmentation to lighten in all populations. However, this scenario assumes stable, long-term maintenance of indoor lifestyles and widespread access to vitamin D supplementation, conditions that may not persist. Conversely, if climate change, resource conflicts, or other global challenges disrupt modern technological societies and push populations toward more outdoor-based lifestyles, UV exposure would increase again, potentially strengthening selection on pigmentation. The direction and strength of future selection on skin color depend on unpredictable social, technological, and environmental trajectories. Advances in genetic technology raise speculative possibilities for directly altering pigmentation. CRISPR and other gene editing technologies theoretically allow for modification of pigmentation genes in human embryos, potentially enabling parents to choose their children's skin color. Such applications raise profound ethical questions and are currently prohibited in most jurisdictions. The medical, social, and ethical implications of genetic editing for pigmentation or other traits remain subjects of intense debate. One interesting question involves the timescale of evolutionary change. How quickly could human pigmentation change in response to new selective pressures? The answer depends on several factors: the strength of selection, the genetic architecture of the trait, population size, and generation time. Historical evidence suggests that pigmentation can evolve relatively quickly when selection is strong. The evolution of light skin in Europeans appears to have occurred within 10,000 to 30,000 years, possibly much faster in some populations. Given that human generation time is approximately 25 to 30 years, this represents roughly 300 to 1,200 generations. Genetic studies showing evidence of ongoing selection on pigmentation genes in recent millennia suggest that significant changes can occur on timescales of thousands of years when selection is strong. However, relaxed selection or weak selection produces much slower change. If modern interventions substantially reduce health consequences of pigmentation mismatches with UV environment, evolution toward new pigmentation optimum would be extremely slow. It might take tens of thousands to hundreds of thousands of years to see substantial change under weak selection. Population size also matters. Larger populations contain more genetic variation and experience more mutations, providing more raw material for selection to act upon. However, the selective advantage of beneficial variants spreads more slowly in larger populations because new variants represent smaller fractions of the total population. For global human populations numbering in the billions, evolutionary change through natural selection alone would be extremely slow. Gene flow complicates predictions about future evolution. Even if strong selection favors particular pigmentation in specific regions, migration of individuals with different pigmentation can counteract local adaptation. If gene flow between populations is extensive, local adaptation becomes difficult because advantageous genetic variants in one environment keep being diluted by variants from populations adapted to different environments. Looking at the very long-term future, hundreds of thousands to millions of years, human pigmentation evolution becomes even more speculative. If humanity colonizes other planets or orbital habitats with different UV environments, populations might adapt to these new conditions. Space environments might have no UV exposure without artificial sources, potentially leading to evolution toward very light skin to maximize vitamin D synthesis from limited UV. Conversely, settlements on planets with intense UV, perhaps due to thin atmospheres, might see evolution toward enhanced pigmentation. However, such long-term scenarios depend on technological and social developments that are impossible to predict. Advances in genetic engineering might allow for rapid, deliberate adaptation to new environments rather than waiting for natural selection. Alternatively, humanity might modify environments to suit current biology rather than modifying biology to suit new environments. One certainty is that skin color will remain a continuously variable trait. Even if global populations mix extensively, creating more intermediate and variable pigmentation worldwide, the polygenic nature of the trait ensures that substantial variation persists. Multiple genes contribute to pigmentation, and each individual inherits different combinations of variants, ensuring that phenotypic diversity continues across generations even within interbreeding populations. The study of recent and ongoing human evolution, including changes in pigmentation, provides insights into human adaptability and evolutionary dynamics in rapidly changing environments. Humans face unprecedented environmental changes due to technology, medicine, lifestyle shifts, and climate change. Understanding how evolutionary processes interact with these changes helps us anticipate health challenges, appreciate human diversity, and make informed decisions about public health and medical interventions. In contemplating the future evolution of skin color, we must remember that evolution has no foresight or direction. It does not progress toward any predetermined goal but simply responds to immediate selective pressures through differential survival and reproduction. Whatever pigmentation patterns exist in future human populations will reflect the interplay of selection, mutation, gene flow, and drift acting on the genetic variation present in contemporary populations. The trajectory is unpredictable, contingent on social, environmental, and technological developments that we cannot foresee. What we can say with confidence is that human skin color, like all traits, remains subject to evolutionary forces. Natural selection continues to act, though its direction and strength may differ from the past. Gene flow continues to reshape global variation patterns. Mutation continues to generate new genetic variation. These processes ensure that human pigmentation, far from being fixed and immutable, remains dynamic and responsive to changing circumstances, just as it has been throughout our evolutionary history. And so, dear listener, we have traveled together through time and across continents, exploring the magnificent story of human skin color. From the sun-drenched savannas of Africa where our species first emerged, through the great migrations that carried humanity to every corner of the earth, to the modern world where we grapple with both the health implications and social meanings of pigmentation diversity, we have witnessed evolution in action, shaping this most visible of human traits in response to the invisible force of ultraviolet radiation. What have we learned from this journey? We have learned that human skin color is not arbitrary or meaningless, but represents a finely tuned adaptation to the UV environment of our ancestors' homelands. We have learned that the same evolutionary process, natural selection balancing the competing demands of vitamin D synthesis and folate protection, has shaped pigmentation in populations worldwide, producing a predictable pattern of variation closely matching global UV distribution. We have learned that this single trait involves the coordinated action of dozens of genes, each contributing incrementally to the final phenotype, creating the beautiful spectrum of human pigmentation we observe today. We have learned that skin color evolved rapidly, on evolutionary timescales, when human populations encountered new UV environments. Light skin in Europeans and East Asians emerged within the past 10,000 to 30,000 years, demonstrating that traits that appear fixed and ancient may actually be recent evolutionary innovations. We have learned that similar adaptive challenges produced similar solutions through different genetic pathways, with Europeans and East Asians independently evolving lighter skin through convergent evolution, modifying different genes to achieve similar phenotypes. We have learned that skin color represents an exquisite example of evolutionary trade-offs. There is no universally optimal pigmentation, but rather different optima for different environments, each balancing protection against UV damage with the need for adequate vitamin D synthesis. The darkest skin protects folate and prevents cancer in high UV environments but risks vitamin D deficiency in low UV regions. The lightest skin maximizes vitamin D synthesis in weak sunlight but burns easily and increases cancer risk in intense UV. Intermediate and variable pigmentation, with strong tanning ability, provides flexibility for populations experiencing seasonal variation in UV intensity. We have learned that the genes controlling pigmentation represent a tiny fraction of the human genome, and that the genetic differences between populations in pigmentation-related genes are exceptional precisely because they have been strongly shaped by local environmental adaptation. The vast majority of human genetic diversity exists within populations rather than between them, and pigmentation provides no information about intelligence, behavior, personality, or any other complex traits. We have learned that modern circumstances create health challenges related to skin color. Dark-skinned individuals in low UV regions face vitamin D deficiency risks unless they supplement or take special measures to maximize UV exposure. Light-skinned individuals in high UV regions face elevated skin cancer risks unless they practice sun protection. These challenges reflect mismatches between our evolved biology and current living conditions, requiring medical and public health interventions to maintain health. Most importantly, perhaps, we have learned that understanding the evolutionary biology of skin color thoroughly undermines racist ideologies that have caused immeasurable suffering throughout history. The scientific evidence is clear. Skin color is an environmental adaptation affecting a small number of genes, varying continuously rather than falling into discrete categories, providing no basis for racial hierarchies or classifications, and carrying no information about cognitive abilities, moral character, or human worth. The diversity of human pigmentation reflects the diversity of environments our species has inhabited, testifying to human adaptability and the power of natural selection to shape organisms to their circumstances. As you drift towards sleep, consider the wonder of this story. Every human being alive today carries in their skin a record of their ancestors' environments, written in the language of melanin and genes. The amount and type of pigment your melanocytes produce reflects thousands of years of evolution in specific UV conditions, balancing competing biological demands with exquisite precision. Your skin color, whatever it may be, represents an evolutionary solution to environmental challenges, a functional adaptation honed by countless generations of natural selection. Consider too the unity revealed by this diversity. While humans display a remarkable range of pigmentation, from the deepest browns approaching black to the palest creams and pinks, these differences emerged recently in evolutionary terms from a common ancestral dark pigmentation. We are one species, one family, shaped by shared evolutionary history and differentiated only superficially by adaptation to varied environments. The genes that differ between populations are few, affecting mostly pigmentation and a handful of other visible traits, while the vast majority of our genome remains shared, testament to our common origin and continued intermixing. The story of skin color teaches us about evolution, how natural selection shapes organisms to fit their environments, how traits that appear fixed may actually be recent adaptations, how similar challenges produce convergent solutions, and how evolution has no foresight but simply responds to immediate pressures through differential survival and reproduction. It teaches us about human diversity, that visible differences reflect environmental adaptation rather than fundamental biological divisions, that socially constructed categories of race do not correspond to meaningful genetic boundaries, and that understanding our evolutionary history can help combat harmful ideologies built on superficial appearances. It teaches us about the complex interplay between biology, environment, and culture, how human innovations like clothing, shelter, and dietary practices have influenced our evolution, how modern circumstances create new health challenges by mismatching our evolved biology with current conditions, and how our understanding of ourselves continues to deepen as science reveals the mechanisms underlying human variation. And it teaches us about the ongoing nature of evolution. Humans are not finished products but organisms continuing to evolve in response to changing circumstances. Our pigmentation today reflects past environments, but future changes in UV exposure, lifestyle, migration patterns, and selection pressures will continue to shape human skin color in the centuries, millennia, and millions of years ahead, just as they have throughout our past. As your breathing slows and deepens, as your body settles into rest, appreciate the evolutionary marvel that is your skin. This living organ, constantly renewing itself, protecting you from the environment while allowing essential interactions with sunlight, carries within it the story of your ancestors' journeys. From Africa to wherever your forebears eventually settled, from the specific UV environments they inhabited to the genetic variants they accumulated and passed down through generations, your skin tells a story millions of years in the making. Sleep well, knowing that you are part of this grand evolutionary narrative, this magnificent example of biological adaptation to environmental diversity. Your skin color, like that of every human being, represents evolution's elegant solution to the challenge of balancing photoprotection with vitamin D synthesis, a solution tuned precisely to the UV environment of your ancestors' homeland. In this understanding, we find both scientific insight and a deeper appreciation for the unity underlying human diversity, the shared evolutionary journey that connects all of us despite our superficial differences. If you enjoyed learning about the fascinating evolution of human skin color tonight, please consider liking this video and subscribing to our channel for more explorations of history, science, and the remarkable story of humanity. Your support helps us continue creating content that educates, enlightens, and helps you drift peacefully into restful sleep while expanding your understanding of the world. Sleep well, and until next time, may your dreams be filled with wonder at the beautiful complexity of human evolution.